composed of a phosphate group and a 5-carbon sugar and nitrogen in spaces. DNA has 4 bases called adenine, thymine, cytosine, and guanine. Adenine only pairs with thymine, and cytosine only binds with guanine. In RNA, uracil replaces thymine and pairs with adenine.
DNA is separated into 2 single strands, creating a replication bubble. DNA helicase is an enzyme that breaks apart base pairs. It disrupts hydrogen bonds between base pairs to separate the strands into a Y shape, known as the replication fork. This area will be the template for replication to begin.
DNA has 5 and 3 end. The 5 end has a phosphate group, and the 3 end has a hydroxyl group. The 2 strands, leading and lagging strand, are antiparallel. The replication occurs in the 5 to 3 direction. Leading strand goes from 3 to 5 direction, while the lagging strand goes from 5 to 3. The 2 sides are replicated with 2 different processes to accommodate the directional difference.
The single-strand binding protein stabilizes the unwound template strands and prevents the 2 strands of DNA from fusing together. In the leading strand, a primase binds to create a short piece of RNA called a primer. It uses the parental DNA as a template. After the RNA primer is created, DNA polymerase III synthesizes continuously in 5 to 3 direction as the fork progresses. Then DNA polymerase I replaces the RNA to DNA. Phosphodiester bond is formed between 3-carbon atom of one sugar molecule and the 5-carbon atom of another.
Because the lagging strand runs in 3 to 5 direction, it begins replication by binding with multiple primers. The replication is broken down into several segments. Primase binds to create a short piece of RNA primer. Then the DNA polymerase III joins RNA nucleotides into a primer and adds a piece of DNA to the strand in between primers. DNA polymerase III detaches when it reaches to the next RNA primer.
Again, primase binds and creates an RNA primer next to the first DNA fragment. Then the DNA polymerase III binds next to the second RNA primer and creates a second DNA fragment. The 2 DNA strands next to the RNA primers are called Okazaki fragments. DNA polymerase I comes and replaces RNA with DNA. Finally, DNA ligase joins the Okazaki fragments together with a phosphodiester bond to form a single strand. At the end of the process, 2 DNA strands are produced, each with 1 strand from the parent strand and with 1 new strand.